Welcome to the deep dive. Today, we've got a really specific mission. We're taking one pretty technical research paper and basically breaking it all down for you. We want to get you past the abstract, skip the really dense methods, so you can walk away feeling like you really get this scientific breakthrough. The paper we're digging into today came out in September 2023. It's called Environmental DNA Reveals the Genetic Diversity and Population Structure of an Invasive Species in the Laurentian Great Lakes.
That's the one. And this study, Andrés is the lead author, it really is a milestone. If you think about the big picture, eDNA, environmental DNA, it's been amazing for answering, a simple question. Is this species here? Yes or no? But this paper asked something much harder to ask. Can eDNA give us that really detailed, fine-scale genetic information, enough to see the structure within a population, not just if it's there?
Right, that's the big leap, isn't it? We know eDNA, the bits of skin, waste, whatever organisms shed into the water, it's great for just detecting species. But if you want to know, how is it moving around? Are there distinct groups? Basically the high-resolution genetic picture, well, you need more complex genetic markers for that. So the core question they tackled was, well, it sounds simple, but it's technically really tough. Can we reliably use eDNA to get at the nuclear DNA, the new DNA? Because that's where most of the really complex genetic info is stored. And getting that level of detail from eDNA, that's been a major roadblock for a while. Okay, so let's unpack why it's been such a challenge.
Yeah, absolutely. We should start with, why eDNA was so successful in the first place. It's non-invasive, super efficient. It really did change biodiversity assessments. You collect a water sample instead of, say, netting fish for days. And with things like metabar coding, you can potentially spot hundreds of species at once, often finding things traditional methods miss.
But then, the population geneticists try to use it to see differences within one species, and they hit that wall you mentioned. Mostly, they had to fall back on mitochondrial DNA, mtDNA.
Exactly. And mtDNA has, limitations for these really detailed population studies.
Right.
Mitochondria, they're the cell's power plants, right? Their DNA is usually just passed down from the mother. It acts like a single data point, a single locus. And that's often just not enough information to map out complex stuff like gene flow or how populations are mixing or these fine-scale patterns. To really dig into population genetics, you need to look at variation across multiple independent nuclear genetic markers. These are specific spots on the main chromosomes. And in this study, they focused specifically on something called microsatellites.
Okay, microsatellites. For anyone listening who isn't familiar, what are they exactly, and why are they kind of the go-to for this kind of work?
So microsatellites are these short repeating bits of DNA sequence. Think of them like little genetic stutters in the code, like AT. The key thing is the number of repeats varies a lot between different individuals. So they act like really good genetic fingerprints. If you look at several of these microsatellite locations or loci, you can get a really accurate picture of relationships, genetic diversity, how genes are moving between populations, all the things you need to understand, say, an invading species.
And the perfect test subject for pushing this eDNA tech was the invasive round goby, Neogobius melanostomus.
Yes, exactly. The round goby is it's a perfect storm for this kind of study. It's a Eurasian fish, invaded the Great Lakes really fast, first seen in the St. Clair River back in 1990. And because it's had multiple introductions probably and spread so quickly, it actually has a lot of genetic diversity and a known complex population structure already mapped out from traditional studies. So it was the perfect benchmark, really. Can eDNA replicate what we already know from tissues?
So the researchers basically went out with three key questions in mind. First, can we even detect and amplify these nuclear markers from water samples reliably? Second, do the results we get from the eDNA, the allele frequencies, do they actually match what we get from physically sampling the fish tissues? And third, crucially, do the bigger patterns, the population structure maps, do they look consistent between the two methods? So how did they set it up? They went out to 14 sites across the Great Lakes region, and at each spot they collected two kinds of samples. The water for the eDNA and actual fish tissues, up to 28 individual fish per site to get that ground truth genetic baseline, covering the old invasion spots and the newer areas where it's still spreading east.
Right, and this is where they immediately saw that big technical hurdle when they quantified the DNA using qPCR assays. That's quantitative PCR.
Yeah.
It basically lets you count the DNA copies in your sample. Now, they found round goby DNA pretty much everywhere. The mitochondrial DNA, the mtDNA, was in 100% of the samples. The nuclear DNA, new DNA, was in 92%. So far, so good.
Yeah.
But the amount The concentration difference was just huge. The mtDNA was massively more abundant than the new DNA.
Can you give us the number on that disparity? Because I think it really drives home how tricky this experiment was.
Yeah, it's pretty stark, and you'd sort of expect it, theoretically, but seeing it in practice, why? Why? Because each cell has only one nucleus, with its 2 copies of the main genome, the new DNA. But that same cell has hundreds, maybe thousands, of mitochondria, each with its own little circular DNA. So the average ratio they found, get this, 196 copies of mtDNA for every single copy of new DNA floating around in that water.
Wow, 196 to 1. Okay, for those of us not pipetting in a lab, what does that practically mean? When you're trying to find and analyze something that's like almost 200 times rarer than the other related DNA.
It means you're fighting against noise and potential failure at every step, especially for these nuclear markers, which are often single copy genes like the microsatellites they were targeting. With such tiny amounts of starting material, you run a really high risk of what's called PCR stochasticity. That's basically the random chance that the amplification reaction just fails to latch onto and copy the few molecules that are there. And that leads to missing data points or maybe results that aren't quite accurate, especially if you're trying to detect rare genetic versions, rare alleles.
Okay, so that's a massive challenge. How did they actually manage to get reliable data despite that huge concentration gap? They were quite clever and very rigorous about it. First, they used that panel of nuclear microsatellite markers, the gold standard ones we talked about. Then, to deal with that randomness, that stochasticity risk from low DNA amounts, they didn't just run the amplification once per sample, they ran it in triplicate, 3 separate times for each water sample. By doing that, they could cross-check the results. If an allele popped up consistently in 2 or 3 of the runs, they could be much more confident it was real and not just a random fluke of the PCR.
Ah, okay, like a built-in error check. And they also had that filtering step, right, to make sure they were only looking at round goby DNA.
Yes, absolutely critical. They compared all the genetic variants, the alleles they found in the eDNA, against the known alleles from the round goby tissue samples. Anything that didn't look like a known round goby allele got filtered out. This was super important to avoid false positives and also to prevent accidentally amplifying DNA from other similar fish, like the tuberose goby, which lives in the same areas and could confuse the results.
Okay, so that careful approach, the triplicates, the filtering, it seems like it really paid off when they looked at the results. Despite that 196 to 1 disadvantage, the eDNA actually did a surprisingly good job.
It really did. They had great success with allele recovery. Overall, the eDNA managed to detect 66.8%. So about 2/3 of all the different alleles they knew existed from genotyping the actual fish tissues. That's pretty remarkable given how little new DNA they started with.
And it got even better for the more common variants, right?
Exactly. When they looked just at the alleles that were reasonably common in the fish present in more than 10% of the tissue samples, the eDNA recovery rate jumped up to 83.7%. That really suggests the eDNA is reliably picking up the main genetic signals, the dominant patterns within the population.
Okay, but recovery is one thing. Did the frequencies match up? Like if an allele was common in the fish tissues at one site, was it also common in the eDNA from that same site?
Yes, and this was maybe the most crucial finding for validating the method. They found a positive correlation. The allele frequencies they estimated from the eDNA RID counts lined up pretty well with the frequencies from the tissue samples. The overall correlation coefficient was R equals 0.69, which, isn't perfect, but it's a solid positive relationship across all the different sites they sampled.
And this is where it gets really interesting, I think, moving from just technical success to actual ecological insight.
Okay.
Because both types of data, the eDNA and the traditional tissue samples, they painted the same picture of how the populations were structured, right?
That's right. They showed congruent patterns of genetic differentiation. What that means is, whether you use the eDNA data or the tissue data, you saw the same basic map of which populations were similar to each other and which were distinct. Specifically, both methods confirmed a known pattern for round gobies in the Great Lakes. Isolation by distance. It's a classic concept in genetics.
Right, basically fish populations that are physically farther apart tend to be more genetically different, just because they don't mix as much.
Exactly. And the tissue data showed this clearly, with a correlation of about 0.74 between genetic distance and geographic distance. The eDNA data picked up the same pattern, though the signal is a bit weaker, a correlation of 0.38.
Still, the fact that the eDNA captured that same large-scale structure, that known invasion pattern across the lakes, that's huge.
It absolutely is.
Yeah.
But, we need to be balanced here. It wasn't perfect. While the eDNA nailed the allele frequencies and the overall structure, it did struggle a bit with some of the standard population summary statistics.
Okay, where did it fall short? What couldn't it measure reliably?
Things we use to measure the total amount of genetic diversity within a population. Metrics like allelic richness, just the raw number of different alleles, and also expected heterozygosity, which is a measure of genetic variation.
Yeah.
These tend to be lower in the eDNA samples compared to the tissue samples, so the eDNA wasn't quite capturing the full breadth of all the genetic variants present, especially the rarer ones.
Okay, so it gets the main picture but maybe misses some of the fine details of total diversity.
Right. And weirdly, kind of paradoxically, the number of private alleles those genetic variants found only in one specific population sample was actually higher in the eDNA.
Higher? How does that work?
It's likely an artifact, again, of that low new DNA concentration and the random chance involved in PCR, that stochasticity. You might, just by chance, happen to amplify a very rare allele in one triplicate run from one site's water sample. And because it's rare, it doesn't show up elsewhere. So it looks like a private allele unique to that site. But it might not reflect a true biologically meaningful private allele that's common enough in that population to be consistently detected. It introduces a potential bias.
Ah, I see. So the random detection of very rare things can skew certain metrics. That's a really important nuance. So it seems like we've moved past simple detection. We can get population structure, but maybe not every single population genetic metric is perfectly reliable yet from just a water sample. Still, like the paper says, it's a substantial advancement. We're clearly beyond just relying on that simple mitochondrial DNA marker.
Definitely. And what's fascinating here is thinking about the immediate potential for managing invasive species, like in the Great Lakes. If you can use eDNA, just from water, to get this kind of fine-scale genetic structure, well, that could become a powerful tool, maybe even in near real time. You could potentially identify the geographic source of new introductions much faster. Knowing exactly where newly detected invaders came from helps managers figure out the pathways they're using to spread. And that's critical if you want to try and prevent future introductions or target eradication efforts early on.
Yeah, and you have to think beyond just this one fish, right? This whole non-invasive approach. It's potentially huge for species where getting tissue is hard or risky or just something you really want to avoid. Like imagine trying to get genetic samples from really rare endangered fish way down in the deep ocean.
Right.
Or big elusive animals like whale sharks or bowhead whales, which I think they mentioned in related work. Taking a water sample is way, way easier and less stressful for the animal than, say, darting it for a biopsy.
Absolutely, yeah. But the researchers are also very clear, more work is needed. There are still challenges. Broadly, they fall into two camps, technical and analytical. On the technical side, it still comes back to that low concentration of nuclear DNA. We need ways to get more of it or make better use of what we get. Maybe that means filtering much larger volumes of water, hundreds of liters, instead of just a few. Or maybe it means optimizing the lab steps, the DNA extraction, to be even more efficient, lose less of that precious new DNA. Especially if we want to confidently detect those really rare but potentially important genetic variants.
Makes sense. And the analytical challenges.
Analytically, the core issue is that eDNA is inherently a mix. It's a soup of DNA from potentially hundreds of individual organisms. But traditional population genetics theory, a lot of the models, they're built on having data from individuals. Knowing the genotype of specific fish A, fish B, fish C, we don't get that from a bulk water sample. So we need to keep developing new statistical methods, new computational tools that are specifically designed to work with this kind of mixed bulk eDNA data and still accurately estimate those population parameters.
Right, interpreting the soup. And then there's the scope, I guess. This was one species.
Exactly. This is a big success for the round goby. But the next frontier, which hasn't really been cracked yet for these detailed nuclear markers, is doing this for multiple species at the same time, from the same water sample. Imagine getting population structure maps for a whole community of fish simultaneously. That's the goal.
So pulling it all together, what's the big takeaway here? It feels like eDNA is really evolving. It's moving beyond just being an early warning system, that simple yes-no detection tool. It's becoming this much more sophisticated instrument for conservation and management, giving scientists the power to map out how invaders are structured, how they're moving, maybe even where they came from, across huge places like the Great Lakes, all without having to catch or even see a single fish.
Precisely. And that really leads to a final thought, something maybe for you, the listener, to mull over. If just sampling water can now start to reveal these complex, hidden genetic patterns of migration and structure for an invasive species, using that tricky nuclear DNA, what kind of incredible new doors does that open? Think about using this for non-lethal monitoring, for conserving species that are incredibly vulnerable or rare or live in places we can barely reach, like the deep sea. It suggests that basically every river, every lake, every bit of ocean is carrying this constantly updated genetic history book, and we're just now learning how to read it.